This conference will now be recorded.
Okay, good evening, all of you, and welcome to the crash course for CUET for PG Life Sciences. And this is Somoju Bhargavi, Faculty of Biosetup Life Sciences. And today I am going with the topics of life sciences, and that is mainly I am dealing with the techniques, biochemistry topics, and other two topics also. And today we are going to start with the first topic, that is the techniques topic. And before going to that, I think you know about the paper pattern of this examination, and it consists of mainly part A and part B. And part A, that is consisting of the language comprehension or verbal ability, and that is mostly related to the English language. And next, coming to the general awareness and quantitative ability, analytical skills, and that means reasoning, quantitative aptitude. And these topics are consisting of 25 MCQs. Okay, and next part B of domain knowledge, and that is of your subject topic, and that is of biotechnology. If you are related to microbiology, then microbiology, like that. Okay, and that is consisting of the 75 MCQs. And what is the marking scheme of the examination means here, and that each question it carrying the 4 marks for that, and for each correct response you will get 4 marks, whereas if incorrect means, and you will lose the 1 mark for that. Okay, this is the main marking scheme of the examination. And that means in that the subject mainly occupies the 75 questions, and that means 75 fours, and that consisting of the 300 marks. And that means you have to know about the basic concept of the total syllabus. And that means if you know the answer of that particular question only, then you can answer that question. And that means you have to know the full concept of the total syllabus. Okay, and then I am going with the topics, and in that the first topic is the techniques topic. I think you know about the techniques because all are completed your BSc, and in that you can choose the biotechnology or microbiology. I think you know about the total concept of that syllabus. Okay, in that, here first of all I am discussing with the techniques means here, as a life science student, you have to well know about the techniques that are used in this biotechnology, and that is mainly consisting of the separation or purification, or what we have to do for the extraction of different biochemical molecules or for the separation of different molecules. And that is mainly consisting of the different techniques like chromatography, electrophoresis, and like PCR also is there, blotting techniques are there, and radioactive isotopes are, these are mainly used for the identification purpose, and that may be of qualitative purpose, and that may be of quantitative purpose also. We can check the quality of a particular biochemical compound, or we can estimate the what is the concentration that is present in the particular component. And that means here mainly these techniques are mainly used for the determination of a particular biochemical compound. Okay, after the learning of these techniques we will go with the biochemical compounds, and today I'm going with the chromatographic techniques. Basically, what is chromatography? Sorry, what is basically chromatography? And this is mainly this is a biophysical technique that mainly enables the separation, identification, and purification of the different types of mixtures that are present, and that is mainly used for the qualification and also for the quantification procedure. And who introduced the chromatography techniques? Anyone? Who introduced this chromatography? Any one of you? Who introduced chromatographic techniques? and that is mainly used for the separation of plant pigments. Use the chat box, ma. Please keep the answer in chat box. Anyone? Yeah, okay. Sweat. TSWETT. Sweat first introduced the chromatographic technique for the separation of plant pigments. This chromatography technique that is mainly used for the separation of plant pigments. That means the plant pigments mainly consisting of the chlorophyll components, carotenoids, xanthophyls, like that. Many pigments are present now. In that, here this chromatographic technique is mainly used for the separation purpose. Okay, and then this is mainly used for the qualification and quantification. What is meant by quality and what is meant by quantity? For example, quality is there, and quality means that may consisting of the different type of components, and that means if we are taking some raw material means, and that is consisting of the different materials, and here mainly we are talking with the biochemical components, and that biochemical components that may be proteins or that may be carbohydrates and lipids, like the different type of biochemical components are there, and we have to first of all separate a particular component means, and that may be depending upon the size or depending upon the different charging particles, like that. Here we can separate the particles that are present in the mixture, and that means the qualitative analysis means that is mainly relating with the different aspects for the separation. Whereas the quantity means here we are separating the different type of biochemical components, and we have to know what is the concentration that is present in the mixture. For example, if carbohydrate is present, and what is the quantity that is present, and if protein is present, what is the quantity that is present, and that means we have to do the quantification means we are using this quantitative analysis, and that means this chromatographic technique that is mainly useful for the qualification and quantification processes, and this may be using differences in sizes, binding affinities, charge, and other properties, and based on that here also different type of techniques are present. Okay, and that means it is mainly used for the separation of different components that are present in the mixture. Okay, and next coming to the, and this is mainly based on the nature and structure and size and also on other properties. Just a minute. Okay, and based on that here we are separating the different components means here this is mainly using the two components, two essential components, and that are of stationary phase and mobile phase. Here the stationary phase, and that means stationary means that is a static position that is not moving, and that may be of either solid or liquid particles, and that is attaching to a glass or metal surfaces. Okay, and here we are separating different components of mixture, and that is or absorbing it to a particular component, and that means here what is the adsorbent we are using, and that is of stationary phase. Understood ma? Here stationary means here that is the static position ones, and that is mainly coated onto the surface of glass or metal surface. Okay, and that means it is not moving phase, this is a static phase. And here in this the most substances that we are used as the stationary phase means that may be of porous ones, and that may be of charged ones also, and also that may be of gel beds, like that. Here we are using different type of materials for the separation of different type of particles based on the stationary phase. And this stationary phase, and this is selected for different chromatographic process. I think you know about the different types of chromatography techniques, like paper chromatography is there, thin layer chromatography is there, gel chromatography, and HPLC is also there na? And in these techniques here we are using different type of stationary phases, and depending upon the stationary phase here we are separating the different type of components, and that means the first of all the separation is mainly depending upon the stationary phase. Okay, and depending upon that, here the chromatography, and that may be using gel beads or thin uniform paper or silica ones or glass ones or some gases or even liquid components that may be that are used as the stationary phase. Okay. Just a minute. Okay. Next, here, and the next phase is the mobile phase. Mobile means here that is the moving phase, and that may be either liquid phase or gas, and that is mainly passing through a chromatography system. And that means here we are considering two phases. One is the non-mobile phase, and other one is the mobile phase. In that, the mobile phase is going through the non-mobile phase, and that causes the separation of the mixture, and that is mainly based on the different characteristics. Okay, and here we can see, here you can observe the different phases that are present here. That is the stationary phase and mobile phase. Mobile phase are the moving ones, whereas the stationary phase here the round ones that are mainly of the static ones. Okay, and here the mobile phase, and that is of a solvent type. This is a solvent type, and that is mainly causing this separation. This is the solvent, and that is carrying the mixture, and that causes the separation. And that means here we have to choose the mobile phases also for the separation of different components. And the solvent means the solvent is mainly present in high amounts actually, and in that the solvent we can take alcohol or water, acetic acid, acetone, or some gases that are mainly used as the mobile phases. Okay, and here based on the stationary phase and based on the mobile phases and based on the characteristics of the components we have to separate, and based on these all characteristics here different type of chromatographic techniques are present. And in that, that may be of affinity chromatography, paper chromatography is a thin layer, like that here different type of chromatography. And let we will discuss about the principles and what is the procedure that is mainly involved and what is the use of that particular type of chromatography. Actually, in exam, how these questions arise means here, and that may be in type of matching type, or that may be in type of statement like questions will arise, statement type questions, or directly they can ask the questions, or sometimes they can ask in type of examples. And that means here one example they will give, and what is the type of chromatographic procedure that is mainly used for this separation. And like that here that is mainly asked in the exam pattern. Okay, and let us discuss about the types of chromatographic techniques. In that, the first one is the affinity chromatography. And first of all, what is meant by affinity? What is meant by actually affinity, ma? You have to answer the questions, use the chat box. Affinity means, what is affinity? Bhavani, Mona Lisa, Sucharita, Soumyasri, are you there? Okay, attraction towards the face. Good, Purnima. Okay, and that means here attraction means that occurs in between two phases, and that may be that is in between stationary and mobile phase. And that means attraction should occur means here some type of attractive particles should be present in between two phases. right? And that means what is the principle mainly behind the affinity chromatography means here, and that is mainly based on the affinity towards the stationary phase of the system. And that means here we have to separate the components means, and that components that are present in the mobile phase, and here that mobile phase is passing through the system, and here that causes the attraction onto the stationary phase. That is the main principle behind the affinity. Okay, okay, Ankit, good. Okay. Okay, and here, what are the molecules or what are the components that are mainly used in affinity chromatography? And that means here the special type of molecules that are present in the mobile phase that are attracting towards to the stationary phase. And that means here the ligands or the substrate. I think you know about the enzyme substrate complex. Enzyme will bind with the substrate, and that is forming the enzyme substrate complex. And like that also here the substrates or ligands should be attracted towards to the stationary phase, and that means here that should contain some reactive components or the reactive sites for the binding. Okay, and that is mainly or present on stationary phase. What is the stationary phase here? The stationary phase should contain the binding sites or the reactive sites for the mobile phase containing the mixture. Okay, that is the main principle behind the affinity chromatography. You have to remember that one only, then you can answer. Okay, and here after the components are attaching to the stationary phase means we have to separate the particles, and here mainly this chromatography is mainly used for the separation, and that means we have to separate the particles means here we are using some elution process, and that elution procedure, and that is mainly done by changing the pH or by changing the ionic strength or other conditions. Okay, and that is called as the affinity chromatography. That is the main principle behind the affinity chromatography. You can see here what is happening here. Here some target analytes are there, and that is mainly present in the mobile phase. Here the stationary phase is that the blue color ones are the stationary phase, and that are of immobilized receptors. Here what is happening here, depending upon the shape or depending upon the size or depending upon the reactive site, here the molecules that are present in the mobile phase that are attracting towards to the stationary phase, and after that the binding occurs. Here based on the binding, here we can identify the particular component that is present in the mixture. Okay, and that is the main principle behind the affinity chromatography. And how can we do the affinity chromatography means, what are the steps that are involved in this affinity chromatography means, this is mainly based on the preparation of some column, and that column, that means here that is mainly depending upon the stationary phase and mobile phase, and that means here the stationary phase that is present in the column, and that is mainly done by loading it with the solid supports like agarose or cellulose. Okay, and that means these are the porous particles that are mainly attracting some of the molecules that are present in the mobile phase. Okay, and onto which here the substrate ligand with spacer arm is attached. Why we are using here the spacer arm? What is the main function of this spacer arm in affinity chromatography? This is also one of the type of question. Okay, what is the purpose of this spacer arm? Anyone, can you guess? Can you guess, ma? Why we are using this spacer of in affinity chromatography? Here we are using the stationary phase, and that is coated with the agarose or cellulose. Okay, onto which here we are adding the substrate or ligand with spacer arm. Why we are using this spacer arm in affinity chromatography? Anyone, can you answer it? Okay, I will tell the reason here. To prevent the hindrances or the repulsions between the molecules. Actually, what is happening means here the stationary phase, and that is coated with some agarose particles or the cellulose particles, means here some steric repulsions occurs in between the ligands or in between the stationary phase. To prevent these steric hindrances, here we are using some spacer arm. This spacer arm, and that is mainly consisting of the ligand, that is bonded with the ligand. Understood, all of you? All are getting my points? Say yes or no. Okay. Okay, to prevent the steric hindrances, here we are using this spacer arm. And next here, the mobile phase, and that is mainly poured into the column at a constant rate. And then after the mobile phase is moving, means here that causes the binding. And here the ligand molecules that are binding to that particular phase, and then that can be eluted from the stationary phase by changing the condition. And these are the conditions that are mainly for the elution purpose. Elution means that is mainly based on the pH or maybe ionic strength, or depending upon the particles that has to be separated. Okay, and that is the elution procedure. And next, what are the uses of this affinity chromatography? What are the uses? And that means this mainly in between the attraction between stationary and mobile phase means, and this is mainly used for the separation techniques for the enzymes and also for different type of proteins. Enzymes means how can we separate the enzymes? By binding with the substrate. And also this is used for the antigen antibody reactions, and also to remove different type of impurities that are present in the mixture, and also for the detection of different mutations and nucleotide polymorphisms that are present in the nucleic acids. Okay, and that is the main purpose of this affinity chromatography. Understood, ma? First of all, you have to know the major principle behind the affinity chromatography, and then what is the procedure that is present, and then what are the applications that are mainly concerned with that type of technique. Okay, and next coming to the what are the examples of this affinity chromatography? And here mainly this is used for the purification of E. coli beta galactosidase. This is an enzyme, and that is present in a mixture of proteins by using some of the agarose, and that is para aminophenyl 1 beta D galactopyranozole agarose. This is the stationary phase here. Okay, here this agarose is the stationary phase that is used for the separation of beta galactosidase. Okay, and also this is mainly used for the removal of excess albumin and macroglobulin from the serum albumin here, and that means it is mainly used for the separation of the different type of proteins and separation of enzymes. Okay, that is the main purpose of affinity chromatography. And next coming to the anion exchange chromatography. Okay, and this is mainly concerned with the ion exchange chromatography. Actually, ion exchange chromatography, that means ion exchange. What is happening here? Exchange of ions are taking place in stationary phase and mobile phase, and that causes the separation. And that means here that is mainly based on the cations and also based on the anions. And what are cations and what are anions? Cations are the positive charged molecules, whereas the anions are the negative charged molecules. Here, based on the attraction between this positiveness and negativeness, here the that causes the separation of different type of components present in the mixture. Okay, and that means it is a separation technique, and that is used for the negatively charged molecules. Anion exchange chromatography is used for the separation for the negatively charged molecule. Anions means which charge they are carrying. Anion means they are carrying negative charge, and that means that is used for the negatively charged molecules separation by interaction with which type of molecules? Positively charged in stationary phase. Understood, ma? In anion exchange chromatography, what is the charge of stationary phase? What is the charge of stationary phase?
Negative. Negative.
No, ma. Positive
Positive.
charge.
Positive.
Yeah. Anion means we are separating anions. Here, the stationary phase is the positive ones. Understood? Okay. And what is the principle behind the anion exchange chromatography means? That is mainly based on the attraction. Here, that is mainly based on the attraction of positively charged resin. Here, we are using resin. What is the meaning of resin means? Here this is also consisting of the different type of particles, but these are mainly loaded with the charges, and when we are charges using means that is considered as the resin. Okay, and that is mainly used for the separation of negatively charged molecules. Okay, and that means it is mainly based on the principle of attraction of positively charged resin and negatively charged analyte. You have to remember these words, ma. Positively charged stationary phase means that stationary phase is called as the resin, whereas which molecules are separating means that are called as the analytes. Analyte, that are the negatively charged analyte. Okay, and that means here the exchange of positively charged ions takes place to remove the negatively charged molecules. Due to the attraction between the type of molecules, here the exchange is taking place. Here how we are preparing these phases means, first of all the stationary phase is first coated with the positive charges, and then here the components that are containing different charge are passed through that, and here what is happening, the negative charges will binding with the positive charge. Okay, and that is causing the separation. And here when we are using charges, and that means here we are using some type of solution, and that is called as the buffer. Okay, when we are using charges, I think you know about the pH, and that is the negative logarithm of concentration. Okay, H plus ion concentration, and that means here when we are using different type of ionic separations means every time we use the buffer solution. Here also by using different buffers, here we are separating some anions or cations. Okay, and that is the main concept behind this. Okay, and this is a type of ion exchange chromatography. It's a type of ion exchange chromatography, and that is the anion exchange. You can see here the diagram of that. Here the stationary phase that is coated with the positively charged ones. Here the mixture that is containing the positive ones and negative ones. Here the negative ones are attracted towards the positive ones. Okay, and binding of these negatively charged ones to stationary phase, and that causes the separation of negatively charged amino acids. Okay, and what are the steps means? First of all we have to prepare the stationary phase with the positively charged ones, and then we have to pour the mixture, and in that mixture that containing positively charged ones and negatively charged ones. Here the negatively charged ones are attracted towards to the positively charged ones, and by using this here we can separate the negatively charged particles that are present in the mixtures, and that is called as the analyte mixture. Analyte, negatively charged analytes. Okay, and what are the uses of this anion exchange chromatography means? That is mainly used to separate proteins and amino acids from the mixtures, and also here the negatively charged nucleic acids can be separated, and that are mainly used for the analysis of nucleic acids. Why the nucleic acids are in negatively charged, ma? Why nucleic acids are in negatively charged? Why nucleic acids are in negatively charged?
Due to phosphate.
Yeah, good. Due to the phosphate groups here, the nucleic acids are carrying the negative charge. Okay, and also this method is also used for the water purification, where here the anions are exchanged for hydroxide ions, and also this is used for the separation of different metals. Okay, and that are mainly containing different type of negatively charged complexes. And what are the examples means here? This is mainly used for the separation of these nucleic acids from a mixture, and that is obtained after cell destruction. Okay, and also for the separation of proteins from the mixture, that is obtained from the blood serum or also from the different type of tissues also. Okay, and that is the anion exchange chromatography. And next, coming to the cation exchange chromatography, this is different to that of anion. This is opposite to that of anion exchange chromatography. Here, what is the stationary phase here? The stationary phase is the negatively charged ones. Here in cation exchange, cations means that are carrying the positive charge. And that means here the stationary phase is in the form of negatively charged. Negatively charged stationary phase, and that is in the form of ion exchange resin, and that causes the separation of positively charged molecules. Okay, and that means yeah, the stationary phase is first of all should be coated with the negative charges, where the components of mixture that containing positive charges will bind. Okay, and here, and after that here the cation exchange resin component complex, and that can be removed by using different type of buffers and by using different compositions also. And what are the steps means? First of all, we have to prepare the stationary phase, and then here next, that means the stationary phase, the resin should be prepared, and after that here the mixture should be passed, and here that causes the separation of these positively charged ones. Okay, and that are the main steps involved in cation exchange resin. And what are the uses here? And this cation exchange chromatography, and that is mainly used for the analysis of products that are obtained after hydrolysis of nucleic acids. And actually hydrolysis of nucleic acids means here first of all we have to separate the different components from the crude extracts or the crude tissue samples, means that may be containing of nucleic acids, that may be containing of proteins, like the different components are present and that are carrying positive ones and also negative ones. Here after the hydrolysis of nucleic acids, here the components has to be separated, means this cation exchange chromatography is used. And this can also be used for the separation of metals, and that are mainly forming different type of complexes, and also helps in purification of water, and also used for the analyzing of rocks and other inorganic molecules also. Okay, and next coming to the examples here, this is mainly used for the separation of positively charged lanthanoid ions. I think you know about the periodic table that is present in your chemistry syllabus, and here mainly the lanthanoids are the ones that are mainly carrying the positively charged, and these are called as the metals. Mostly these lanthanoids are belonging to the metals, and that means we had to extract the metals from the earth crust remains, and this cation exchange chromatography is used. And also this is used for the determination of total dissolved salts in natural waters by analyzing the presence of calcium ions. Okay, and that means these calcium ions are mainly carrying the positive charge. And here for the determination of these salts, means here we can detect the calcium ions. Okay, and that is the ion exchange chromatography. First of all, we discussed this affinity chromatography, then about the ion exchange chromatography. And ion exchange, again, that are of anion exchange and cation exchange. And next, coming to the column chromatography. Column chromatography. And based on this column chromatography, only here in ion exchange chromatography, we are preparing the columns. Okay, and what is the main principle and what is the main technique involved in column chromatography means here, this is mainly based on the absorption with the stationary phase. Here mainly the components that are present in the mixture, and that are mainly separated based on adsorption, not absorption ma, adsorption. What is the difference between adsorption and absorption? Ad and ab, ab and ad, adsorption and absorption. If you know the difference, means you can understand easily. Okay, adsorption, ADSORPTION. That means sorption means attracting. Adsorption means here that or mainly attaching to the surfaces only. Whereas in absorption means that or moving into the inner surfaces. And here this column chromatography, this is mainly based on the adsorption with the stationary phase. And that causes a separation of mixtures and that are moving at different speeds when these are passed through a column. Okay, and this is a type of solid liquid chromatography technique. This is a type of solid liquid chromatography technique in which the stationary phase is the solid ones, and that means it is mainly consisting of the agarose or the cellulose ones, that is mainly packed with this stationary material, and that is of the solid material, whereas the mobile phase is a liquid or a gas. Mobile phase is a liquid or gas. You have to frame the table for chromatography techniques. First, what are the type of techniques? What is the stationary phase? And what are the mobile phase? And what is the principle? If you maintain the table means you can easily remember these techniques. Understood, ma? Okay, here in column chromatography, here the stationary phase is the solid ones, whereas the mobile phase is the liquid or the gas ones. Okay, and what is the principle? That is mainly means this is mainly depending upon the adsorption, and that means here depending upon the different affinities with the adsorbent present on the stationary phase. Here the mixture that containing different type of components can be identified or can be separated. Okay, the molecules having higher affinity and that are mainly adsorbed onto the stationary phase for a longer time, and that causes the decreasing of the speed of movement through the column. Okay, whereas here if the molecules are attaching with the lower affinity means they are moving with a faster movement, and thus are allowing the molecules to separate fastly. Understood, ma? Here what is happening? Here first of all we are preparing the stationary phase, and that stationary phase is called as the adsorbent, and that is made with the solid particles, adsorbent. Okay, here and on that the mobile phase is running, and what is the mobile phase here we are using? And that may be of liquid or that may be of gas. Here what is the principle involved? Adsorption. That means in between stationary phase and mobile phase adsorption is occurring. Okay, whereas if higher affinity is occurring in between the stationary and mobile phase means what is happening? The mobile phase containing components or retaining onto the stationary phase, and that means that causes the stopping of their movement. Okay, whereas if no affinity means that causes the moving of the components that are present in the mixture. Understood, ma? If the components that containing faster affinity means they are moving slowly, whereas lower affinity means they are moving fastly. That is the main principle behind the column chromatography. Okay, and here what is the adsorbent that is used is here the adsorbent that is mainly used in column chromatography is the silica gel. Silica gel is mainly used. You can see the picture of this column chromatography. Here, these are like burette model, and I think you know about these burettes that are mainly used in your chemistry labs, chemistry practicals here. And these are like columns, and here first of all we have to prepare the stationary phase, and that should be coated onto the surface of this column, and then we have to adding the sample, and that means the samples are to be loaded, and that causes the separation of samples depending upon the adsorption between the stationary phase and mobile phase. Stronger that causes a stronger interactions and weaker interaction, and the stronger interaction that causes the retaining of the particles, that causes the slow movement, whereas that means weaker interactions means that causes the fast movement of the particles. Okay, and this is the major principle, and that is mainly involved in this column chromatography. Okay, and what are the steps means? First of all, we have to prepare the stationary phase, and then here that should be loaded with the mobile phase that containing different type of mixture, and that causes the separation. Okay, and next here, the molecules that are bound to the column, and then we have to remove by using elution techniques means here, and that are mainly by using polarity here. Elution, and that is mainly based on the polarity, or that means here two techniques are mainly used for this elution in this column chromatography, and that are of isocratic technique and gradient technique. Isocratic means here the same polarity is used for the separation, whereas this gradient technique, and that is mainly used for the different polarities with different samples. Okay, and this technique can be used for the different purposes, and here we can separate the different molecules and by using this column chromatography, and that can also be further analyzed for different purposes also. And what are the uses of column chromatography means? This is mainly used for the separation of impurities and purification of biological mixtures, and also for the isolation of active molecules and metabolites from various samples. And also this is mainly used for the detection of drugs and crude extracts also. Okay, and this column chromatography, what are the examples means here? And this is mainly used for the extraction of pesticides from solid samples of animal origin containing lipids, waxes, and pigments. Okay, this is mainly used for the extraction of pesticide, and also for the synthesis of different peptide analogs, hormonal analogs, and mainly of synthesis of promelinitide, which is analog of amylin, and this is a type of peptide hormone which is used for the treating of diabetes, and also for the purification of bioactive glycolipids, and that are showing antiviral activity towards this herpes simplex virus also. And these are the some of the examples regarding this column chromatography. Okay, and this is mainly used for the analysis purpose, purification purpose, and also for the separation purpose. Okay, and next coming to the flash chromatography. Okay, and this flash chromatography, and this is also a separation technique here. Here mainly the stationary phase that is consisting of the smaller sizes of gel particles, and here and the pressurized gas is used to drive the solvent through the column. That means here this is like similar to the HPLC chromatography, and that means here the pressurized gas is mainly used for the movement of the mobile phase here. That is the solvent, for the movement of the solvent through the column. Okay, understood ma? Here flash means, and that is combining with the gel chromatography and HPLC also. Here we are using the stationary phase of gel particles. Okay, small size gel particles that are used as the stationary phase, and the pressurized gas is used for the driving of this mobile phase through the column. And what is the principle means? This is mainly based on the adsorption only. This is also based on the adsorption, but here the sample, how it is flowing into the column is, that is mainly by applying the pressurized gas, and that causes the efficient process for the separation, and also causes the faster separation. Okay, and that means here the molecules that are binding to the stationary phase, and that is mainly based on the affinity means, here we are using the pressurized gas, and here the lower affinity ones, and that are mainly that are moving fastly when applying this pressurized gas, whereas the high affinity means that are attaining some time, that are retaining onto the this stationary phase for some time, and after this higher pressure, and that can also move. And depending upon the time of this movement, here we can identify the components that are present in the mixture. Okay, that means this is similar to that of column chromatography, but here we are using the pressurized gas for the moment of this mobile phase. Okay, and that is called as the flash chromatography. And here you can see, first of all, the column should be prepared with the silica gel, and that means here we are using some type of gas, and that is and this is a gas ones, and that causes the separation ones. Okay, and you have to remember this is a type of column chromatography, but here the sample or the mobile phase is moving with the help of some pressurized gas. Okay, and next, what is the steps means? First of all, we have to prepare the stationary phase, and here and also the mobile phase is added to the mixture, but here the mobile phase is introduced into the column by passing some pressurized gas. Okay. Okay, and next here, what is the elution procedure? Here also the elution procedure, that is mainly based on the polarity, and that is using Isocratic technique and also gradient technique. Okay, and in that also here we can analyze the molecules that are present in this mixture. And what are the uses means? This is mainly used for the separation, faster separation. We have to separate the component, we have to identify the components that are present in the mixture, means this flash chromatography is mainly used, and this is the more efficient method of separation of components of different mixtures. And also for the removal of impurities that are present in crude extracts of natural and synthetic mixtures. And what are the uses? These are the main uses of flash chromatography. And next, coming to the gas chromatography. And here also this is mainly also depending upon the retention time, and that is also based on the affinity of the molecules. And like that here, column chromatography, based on the column chromatography, this gas chromatography is designed. Here also retention of, based on retention of molecules onto the stationary phase, here the separation of molecules occurs. Okay, and how this occurs means, here the sample, and that may be of liquid or gas, and that is vaporized in the injection port. Here what is happening means, here I will show. Okay, and this is the one, and that is mainly used for the gas chromatography. And that means here the stationary phase is the column packed ones. I think you know about what is stationary phase that is present in column chromatography, and that should be used for the gas chromatography also. Okay, whereas here the mobile phase, and that is mainly is a gaseous phase. Here the mobile phase is a gaseous phase, and that is mainly containing of helium gas, and that is carrying the sample through the column. Understood ma? Here the sample, and that should be injected into the column means here, and that should be in the form of vapor stage, and that is passed through a detector, and that causes the retention of this gas onto the stationary phase, or retention of the liquid particles onto the stationary phase, and that causes the separation. Understood ma? Here what is happening in the gas chromatography? The stationary phase is the normal one, like column chromatography, whereas here the mobile phase is the gaseous one. The mobile phase is the gaseous one, and here, and that is carrying the sample, and this sample, and once that is injected means that is converted into the vapor stage, and that is passed through the detector, and that is mainly used for the determination of the separation of different molecules that are present in the mixture. Okay, and that is the main principle involved in gas chromatography. Understood ma? All of you, you can see here what is happening here, and this is the gas flow regulator is there now, some carrier gas, and that is in the form of mobile ones, and here the sample is injected through the injector port. Here the sample device is different from the mobile phase device. Here the mobile phase, that is passed through the gas flow regulator, and here the sample is injected through the injector port, and that is passed into the column, and that column is already packed with the stationary phase. Okay, and here this mobile phase, and that means the carrier gas, and that is causing the forcing of the sample, and that causes the separation. Here the detectors are mainly used for the detection of the retention time of the particles that are present in the mixture. Okay, understood my all of you? Is it clear for you?
And
Is it? Yeah, tell me,
uh,
ma.
if the retention time is more,
Okay,
like then the
movement is
molecules
slow.
mm,
Slow. Yeah. Okay.
directly
Please use
propose
the
more.
chat box for asking also questions, because
Okay.
that causes some disturbances for the others. Na? Okay. You can use the chat box, all of you. Okay, and here these are the steps, ma. First of all, we have to prepare the column, and also for the preparation of column is, first of all we have to use the stationary phase, and then the sample, and that should be injected into the column, and that should be vaporized in gaseous state, and then it is mixing with the mobile phase, and that causing the separation of the components, and that can be detected by detectors. And that detection, that means here, depending upon the retention of the particles, here the identification occurs in different times, and based on that retention time, here we can identify the molecules that are present in the mixture. And what are the uses means, this is mainly used for the calculation of concentration of different chemicals that are present in the samples, and also for the analysis of pollutants, oil spills, other samples. And also this is used for the forensic science studies to identify different chemicals or different molecules that are present in the mixtures. Okay, that are the main uses. And what are the examples means here? This is mainly used for the drug identifications in athletes urine, dopings in doping test, and this gas chromatography technique is mainly used for the doping test, and that means for performance inducing drug in athletes urine is detected by this gas chromatography. And this is also used for the separation and quantification of drugs in soil and water samples also, and that means here mainly this is used for the drug identifications. Okay, and that is of gas chromatography. And next, coming to the gel filtration chromatography, and this is a type of partition chromatography. And what is meant by partition means here, different type of components, and that are containing different molecular sizes, and that molecular sized components can be separated by using this gel filtration chromatography. Hence it is called as a partition. Partition means that is dividing, and dividing means here how these are dividing the molecules, that is mainly based on the molecular sizes. Okay, and that is called as the gel filtration, gel permeation, or size exclusion chromatography, or molecular sieve chromatography. Okay, and here the molecules are mainly partitioned in between mobile phase and stationary phase, and that causes a separation of the mixture that is mainly based on the relative sizes. Here the stationary phase, and that is of matrix, and that is consisting of the porous polymer, and that means it is containing different sized pores. Bigger sized pores are there, smaller sized pores are there. And depending upon the size of this porous polymer, here different type of particles can be separated. Yeah, sample, and that is injected with the mobile phase, and here the mobile phase that is occupying the poles of the stationary phase. And here if the size of the molecule, and that means that is enough, that is appropriate enough to enter the pores means, and that are binding uh, wholly or partly, and depending upon the size, here the binding occurs in between the stationary phase and mobile phase. Okay, and here molecules with the larger size, and that are entered, that are retained from entering the pores means, and that if the largest sized molecules are present in the mobile phase means, here if these are not permitting to move into the pores means, they can be separated easily. whereas the small sized means and they can be fixed onto the stationary phase, and depending upon the size of the pores here, these are eluting with the mobile phase, and that is called as the gel filtration. That means here the filtration of components occurs based on the size of the particles. Okay, and here you can see here, and this is the stationary phase, and that is containing the porous beads here, and these are the different sized particles here. First of all, this is eluting the larger sized particles, and then this is eluting the smaller sized particles, and that means here the larger sized ones, these are not fitting into the beads, and that means here they can be eluted fastly. Okay, and that is the main principle behind this gel permeation chromatography. And what are the steps means here, the column is filled with the semi permeable porous polymer gel beads, and that means here first of all the stationary phase has to be prepared with the gel. I think you know about the preparation of the gels, in page electrophoresis, and also for the agarose gel electrophoresis we will prepare the gels, and like that here also the stationary phase is prepared with the gel, and that causes the separation of the mobile phase containing different type of particles that are mainly based on the different sized ones. Okay, and here, and the molecules that are binding to the column, that are mainly separated, that means elution process. Here the elution process, that is mainly based on the polarity, and also different conditions that are mainly used for the separation. And what are the uses of this gel permeation chromatography means, this is mainly used for the um for studying of the stability and activity of different molecules of interest. And that means here, for example, if a particular component is there, we have to know the particular activity of that molecules means here this gel permeation chromatography, this size exclusion chromatography is mainly used for that purpose. Okay, and this is mainly used for different purposes. This is mainly used for the nucleic acid separations like DNA, RNA, tRNA, and that is containing different type of bases, and that are mainly separated by using this gel filtration chromatography. And what are the examples means, this is mainly used for the separation of recombinant granulocyte factor, that is of human granulocyte colony stimulating factor, and that is mainly from the inclusion bodies, and this is mainly based on the size exclusion process. Understood here? Recombinant human granulocyte stimulating factor, that is present in inclusion bodies, and that is separated by this this gel filtration chromatography. And here what is the gel filtration chromatography that is used here is the urea gradient size exclusion chromatography, and that is mainly based on the gradients in this gel. Okay, and next here the separation of hen egg lysosome, and that is also separated by using this acrylamide and dextran based gel columns. Okay, and this is the gel filtration chromatography, that is the main principle involved in gel filtration. Okay, I think time is out. Okay, and another more two techniques are there, ma, and tomorrow we will discuss the techniques, and we will continue for the next topic also. And these are the different type of techniques, ma. And first of all, you have to prepare some table like format, like in one column you have to mention what is the type of chromatography, and in the next column you have to mention what is the stationary phase, and then mobile phase, and what is the purpose that is using, and sorry, what is the principle that is using, and what is the purpose that is used. You have to maintain like that, then you can remember easily. Understood, ma, all of you? Is it clear? Say yes or no. Use the chat box, ma. Yeah. Okay. Ganga, Sucharita, Ishika. Good. Tiyasha. Okay. Yeah, Bhavani. Okay, ma. If you have any doubts, you can post in chat box, ma. We will continue in the next class, ma. No doubts? What is elution? Okay, good. Elution means here stationary. We are using two phases now. Stationary phase is there, mobile phase is there. And that means here we are using these stationary and mobile phases for the separation of the particles. And here we are using separation means we are using different type of components for the separation. For example, enzyme is there, substrate is there. Okay, substrate means that is mainly occupying the stationary phase, whereas the enzyme that is present in the mobile phase. After what is happening? This enzyme is binding with the substrate, that is forming the enzyme substrate complex. Okay, and that means this enzyme substrate complex is formed in that particular type of technique. And then we have to separate that once. We have to separate this enzyme substrate complex. And that means we have to separate the components that are binded with each other. And that process is called as the elution process. Elution process. For the separation of the particles is called as the elution process. Understood, ma, Ishika? Separation of components that are present in the mixture, that is called as the elution. Mobile stationary. Yeah, good. Yeah, that is the one, ma. Okay. Yeah, I will send the slides, ma. Okay, no issue. I will share the material with you. Tomorrow I will ask the questions related to these. Okay? Yeah, okay.